Hello, in this video I would like to talk about what makes biodigestion possible. Methanogens. I have thoughts about methanogens, I have ramblings about methanogens, and in Biogas USA fashion I have questions about methanogens. So if you Google what are methanogens, it'll tell you they are microorganisms that produce methane, CH4, as a product of their metabolism. If you dig a little deeper, it'll say, these microbes and their exotic metabolism have inspired decades of microbial physiology research that continues to push the boundary of what we know about how microbes conserve energy and grow. So as biogas enthusiasts, I guess you could kind of call us microbial physiologists. I'm gonna add that to my LinkedIn. This video was inspired by actually a viewer comment, like many of my videos. This viewer goes by the name 443DM, or Delta Mike. And his comment reads, CO2 is a byproduct of anaerobic digestion. Minimizing the amount of oxygen gas that gets into your fermentation vessel will push it more in favor of anaerobic digestion with methane as a product instead. I wonder if a layer of cooking oil floating on top would improve this. For some reason, I had not ever thought of. I've been mostly focused on filtering and purifying the methane on the back end after it's produced. I had never thought of nurturing the methanogens that produce it to see if maybe they would do a better job or something. So that's a really interesting thought, and it kind of exploded into a lot of thoughts. So let's walk through it together from the beginning. We all know that when you inoculate a new digester with animal dung, it takes like 2 or 3 weeks before you get anything flammable. And in that time, you make mostly CO2. What's the reason for that? I think it's because there's a diverse array of bugs and microbes and bacteria that you just introduce to that bucket or that barrel. And a lot of them are, they need oxygen and they produce CO2. So that slurry going in is super oxygenated from being mixed up and dumped in and everything. It's going to have a lot of oxygen in it, a lot of air. So I think until that oxygen gets used up and consumed by them, the methanogens are not the dominant party. So after the oxygen gets used up, then the methanogens can kind of start to thrive and take over, and you start producing methane. I think this is also why biodigesters are known to mature after a year or two of operation, because the other cultures, the other bugs in there that are competing for resources, start to die out and become fuel for our friends, the methanogens. So after that, there's less competition for resources, and the results show in the quality of your product and the efficiency of your digester. So to the point of the comment that inspired the video, I'm not sure if a layer of cooking oil would actually reduce the oxygen being introduced. It's not exposed to very much oxygen except for while I'm feeding. While I'm feeding, I'm pouring in new substrate and it's splashing and bringing oxygen and oxygenating the slurry with it. So maybe a layer of cooking oil would create a softer landing. What I did look into was the volume of my feeding tube, the 2-inch PVC going into my digester. I calculated the volume of that to make sure that I'm never feeding more than the volume of that tube in one feeding. My thought there is that the oxygenated liquid would hang out in the feeding tube, and the oxygen would either bubble out, gas off or be consumed by those other bugs, and then the next time I feed, it'll be pushed into the rest of the digester. Those are my ideas there. I don't know if that would help. I calculated the volume of that tube to be 2 gallons though, just under 2 gallons, which I would rarely feed that much in one feeding. So I thought I'd bring it up anyways though, in case it spurs on some new ideas in any of you. Let's see what's next. With this, I'm mostly out of ideas on how to limit O2 in my biodigester, but I'm not out of ideas on how to nurture our friends, the methanogens. So I researched quite a bit deeper to learn about them, and I want to share just a little bit because I found them to be fascinating. They're not actually bacteria. They are archaea, which is its own domain separate from bacteria, which is theorized to be actually closer to eukaryotes like us. Many archaea live in extreme environments like thermal vents and high acid places, high base, high salt concentrations. They probably like the extreme H2S levels inside of my biodigester. So anyways, methanogenic archaea could be a good key term for further research. Also, key terms I found, if you want to learn more about methanogenic archaea, is methanogenesis or biomethanation. Those two words, there wasn't a lot on YouTube. I think it's a pretty understudied category in biology. But I think if you use those words, looking for research papers, you could probably learn a lot. In addition to oxygen, we know that they dislike cold and light. So if painting your digester black helps your methanogens by keeping the light out, then I'll bet you wrapping your digester in a space blanket or tinfoil would help even more. I'll be adding that oil to see if it helps, and wrapping the digester in a space blanket and adding a heater. I'll be doing all that this summer when the production picks up, so I can see if I'll do it one at a time so I can see if I notice any difference in the CO2 levels of my end product. I'll also be implementing whatever other ideas you guys come up with that, I think might work. So my mission here has never changed. My mission has always been to make biogas pure enough to be pipe gas, or at least pure enough to run a car. And I understand, make no mistake, I understand that's been done. These problems have been solved. There are biodigestion plants all over the world currently shipping gas for process, and their product is indistinguishable from well gas, to my understanding. What I'm trying to do here is recreate their plant on a miniature level using mostly widely household available supplies. It's probably not possible. I have thought about hiring like a consultant to sell me the chemical that I need to make pure gas. And I may still go down that route. That might be fun, but I'm not done trying, household supplies yet. Okay, so this is where I'm gonna get really crazy. What if there is no other bacteria, no other bugs in your barrel? If you take your digester and fill it with a nutritious substrate like light malt extract, and you put it on a giant double boiler / ghetto campfire setup, bring the whole thing to a boil, right? Sterilize the barrel. And then as it cools, I guess you'd have to cover a hole in the top with micropore tape so that it doesn't suck in new contaminants as it cools or whatever from the air. And then you'd have a sterile barrel. After it cools completely, maybe you could inoculate it with a liquid culture of methanogenic archaea. I don't know if you can buy a liquid culture of methanogenic archaea, but maybe I could make one. If I can't buy one, maybe I could isolate the methanogens on a plate of agar after I swab some fresh cow poop and culture that. Is that insane? Is that possible? I have no idea. Does it even matter? That's the bigger question. If I were to do that and there is only one bug, only methanogenic archaea in my barrel of substrate, would there be any CO2 at all? Would I produce 100% methane? That is a question. If it was like 90 to 100% methane, this would be probably not even that insane. It would probably actually be worth it. And I may try culturing a swab of cow poop on some agar plates later, just to see what happens. Okay, last thing. There are a lot of very smart people that watch these and engage with them for some reason. I'm not sure why you guys all humor my ramblings and mostly coherent inquiries, but I sure do appreciate it. I get a lot out of these comments. I learn a lot. They stimulate a lot of more ridiculous thoughts, like this entire video. So I appreciate you guys. Thanks for watching. Till next time.